I can bring your guys' window up and I can see all your videos, and it's much less like I'm talking to a black, a black hole. So in fact, the more of you that can turn on your videos, the happier I will be, because I will see some kind of facial expression, which, trust me, is really worth it when you're giving presentations. And so I would greatly appreciate.
I think that the screen went off.
Yeah, it went back.
I see it now. Yeah. Okay.
Yeah,
So
right.
just let me give the formal introduction.
So did the slide advance just to check?
Yes. Yes.
Okay, great. Let's go back. Okay, perfect.
So before I formally introduce today's speaker, I'd like to remind the audience, both at YouTube and here at Zoom, that you may type your questions, and then I will relay them to Joshua throughout the colloquium as he prefers.
Perfect.
So as for the proper introduction, it is a great honor and privilege to have Professor Joshua Shaevitz as our speaker in the ICTP-SAIFR IFT-UNESP Physics Discussion Colloquium Series this week. Professor Joshua is a professor of physics and genomics at the Lewis Sigler Institute at Princeton University, and he's the co-director of the National Science Foundation Physics Frontier Center for the Physics of Biological Function. He got his PhD from Stanford University in 2004 and has held positions at Berkeley, Boulder, and at the Howard Hughes Medical Institute. He has made fundamental contributions to the understanding of the mechanics and dynamics of biological systems, from single molecules to cell collectives to behaving animals, by developing new techniques for precision measurement. And for that, he was elected fellow of the American Physical Society. Today, he will talk about self-driven phase transitions in living matter. So, Professor Joshua, the stage is yours.
Excellent. Thank you very much. This is exciting. Please interrupt and keep your videos on. I can see your eyes. So when you fall asleep, I'll know, and that's on me, not on you. So, okay. All right. So I'm going to talk about a lot of things today. There's different sorts of talks I can give to these kinds of audiences, but this one is sort of a compendium of things that have been going on for the last 10 or 15 years and have involved collaborations between my group and a number of different groups worldwide, including Cam Mignot, who's in Marseille. Manon is now in Germany. Ned is at Princeton. Christina was at Syracuse, but is now in Santa Barbara. Roy is at Syracuse. So this is really, a compilation of work by a lot of different PIs and a lot of different students and postdocs who have been advised, many of them jointly by myself and my collaborators. And it's been a lot of fun. I believe that collaborative research is just more fun than non-collaborative research. I would much rather talk with other people about what we're doing every day than live in my own little silo. So this represents some of that work. Okay, so some of you will know this movie. Some of you maybe not, but I want you to imagine this liquid material here. And it's going to have some special properties that are different than most, like it kind of looks like mercury, right? But it's going to do some things that I don't think you've ever seen a conventional liquid or a conventional material just sort of do. It sort of aggregates on its own, and then it is able to do this amazing pattern formation process where it forms a human-like terminator. And so this is science fiction, right, in some sense, except that this very kind of thing happens all the time in biology, right? The fact that small constituents, whether they be molecules or cells, are able to form complex dynamical patterns all the way up to forming your own body, right, is something that biology knows how to do. And you may have heard, maybe even in this series, about some of the strategies that living organisms use to, for example, form your body plan, where your neck is and your fingers and all this. And almost all of that comes from a mechanism called reaction diffusion. which in its sort of most basic form is if you have a diffusing molecule with a diffusion coefficient, and if that molecule encodes a time scale, for example, if it's has some kind of catalytic rate or some chemical rate associated with it, you can combine that time scale and the diffusion coefficient to make a length scale. And so that process of pattern formation, the wavelengths or the characteristic lengths that you get in reaction diffusion come from those two things: a time scale, usually a chemical time scale, and a diffusion coefficient. And you of course can make this more complicated, but that's the general idea. And that's why you have 5 fingers and you have a neck where it's supposed to be. And maybe you've heard, I don't know, Professor Spreger or Bialik maybe have come and talked about that. Okay, so there are other ways that you can form patterns, and one is really by self-assembly, and through essentially fluid dynamic like instabilities in active materials. And so this is inherently not a reaction diffusion. We're going to have to understand the physics of active materials. And by active, I mean materials in which thermal energy is not the ultimate driving force of the constituent parts of the material. So we're going to be talking a lot about essentially a fluid made of cells, and the cells move themselves around. They do it by burning chemical energy that involves thermal energy. That's fine. But what the constituent, molecules, which are the cells, just run around by themselves. And because they do that, they can steer where they go. They can do lots of things that allow a material made of those kinds of parts to do things that a normal material can't do. Okay, and so we want to try to understand the physics of these active materials, or active matter, it's sometimes called. Okay, so here is the basic process that I'm going to talk about. This is a colony of E. coli. Okay, I'm sure many of you have heard of E. coli. It's a single-celled bacterium. It can make you sick sometimes. This one will not make you sick. You probably all think that E. coli move around a lot, and with this wonderful flagella you may have heard of, and there's lots of interesting things about flagella and chemotaxis and all this. You might be sad to know that E. coli almost never has a flagellus. They only express flagella when they're starving. If they're happy, like if you grow them in a happy test tube, they don't have any flagella. They just sit there and eat all day, right? They eat and divide and eat and divide. They're totally fine. Okay. And then only if they're hungry, they make a flagellum. And they can only move in a liquid environment because they have a fluid dynamic propeller. I have put them on a surface here with essentially no fluid around them. There's a tiny layer of fluid that's wetted into this gel that they're sitting on. And so they can't swim. So they are stuck. So, if you study bacterial motility all your life and you think that flagellar motors are important, well, these guys are in trouble. This, however, is a colony of the cell that we're going to be talking the whole time about, called Myxococcus xanthus. And it's a single-celled organism, just like E. coli, but it doesn't swim, it crawls on surfaces. And not only does it crawl on surfaces, but it eats other bacteria. So it's going to prey on the E. coli. It does this by excreting small molecule antibiotics that end up killing the E. coli. The E. coli essentially explode, and then the Myxococcus just absorb whatever happens to come out of the E. coli. And there's a lot of interesting things about that, the ecology of that. So as we play this movie, what we're going to see is the E. coli or the Myxococcus are going to crawl out over the E. coli. You'll see in this regime, they're actually going to make some really interesting wave-like patterns that allow them to mix themselves over the E. coli and eat them. But I want you to focus on the middle here. Now, these are cells, Myxococcus cells. Yeah, so now you can see the waves are really amazing. Oh, they're fantastic. These Myxococcus cells were not lucky enough to have ever found the E. coli. Okay, so they're actually starving. And what Myxococcus do when they starve is they do something called sporulation, where they essentially go from a long, flexible rod-shaped shell. They collapse themselves into a small sphere. They then coat this sphere with a really protective, hard, rigid shell. And that shell, they can live sort of dormant for long, long periods before food comes again. And then they can wake up, called germination, and crawl out of the shell and go. The problem is the process via which they predate on other cells, which they eat other species, involves many of them. A single Myxococcus xanthus cannot excrete enough antibiotic to kill anything. And so they need to hunt in large groups. And in that sense, we would call them obligately social. They need to be with their friends. So if they sporulate by themselves, if like there's one Myxococcus spore somewhere, it will die no matter what. Because even if it wakes up, it can't make enough antibiotic to kill anything, and then it's just going to die in the end. So what the cells want to do is make a group of spores all together, so that when they finally do detect some prey, when they germinate, there's many of them, and then they can go hunt and do their amazing wave stuff and things like that. Okay. And so what these cells have done, these little black objects are actually three-dimensional droplets. They have dewetted from the surface and formed fluid droplets, and then inside the droplets, they all sporulate. And that's the process that we're going to talk about essentially for the whole time, is this dewetting process. Okay. I did want to show you really quick first is the wave stuff is really amazing. They make really standing waves. So these are different, each black line here is a different time point. You can see there's nodes and antinodes. I mean, there's really fascinating stuff going on with this wave-like instability. We are just starting to get a handle on the physics behind it. They're surface waves, like ocean waves in some sense. I won't talk about that today, but I know physicists like wave kind of phenomena, so I thought I'd show you this pretty picture. You can even get standing waves in bacterial populations and things like this. Okay. So these fruiting bodies, these droplets really are three-dimensional droplets. So this is a three-dimensional picture. It's probably 50 or 60 microns on a side, 20 or 30 microns tall. You can see it has essentially a finite contact angle. At this point, it's round. There's a real surface tension. And so this is a fluid made of living cells, right? So it's not like they're dissolved in a solvent. The fluid itself is made of living cells. It's like if you've ever seen one of those really large ant colonies that's like going over the path in the woods, and it looks like a fluid and it's kind of flowing and all that, right? That's a fluid of ants, right? And no solvent. So this is a fluid of cells. It's very much similar. And they're going to go from this wetted phase where they're kind of on the surface into a dewetted phase, just like water beads up on your raincoat or on a piece of glass, things like that. Okay. This, however, is a single Myxococcus xanthus. So this fluid is made up of millions of these guys. They're small. They're a few microns on a side. They're flexible. They crawl on surfaces, as you can see here. I spent a number of years trying to understand the motors inside of them. This is where I started with this organism. And in fact, why did that go so fast? Go back. They have two different kinds of motors. One is they essentially have little feet that they can move from one side of the cell to the other side of the cell, and they stick those to the ground, and that drags the cell forwards. And the other kind of way they have of moving is with these long grappling hooks. So they extrude polymers that bind to the outside, and they reel them back in from the inside, and that pulls them along like a fishing rod or a grappling hook. And we've done some work recently trying to understand the details of these motors and how they decide which motors are going to make the polymers and all that sort of thing. So there's some nice recent work kind of on the molecular imaging side of that. You don't need to know about either one of these really for this talk, except to know that the cells can move themselves. They have a way of moving on surfaces. The other thing they have is they have two gears. They have a forward and a reverse, although they're the same. So really it's one gear. They just move the gearbox from one side of the cell to the other side of the cell. It's really quite fantastic on the molecular scale. But the consequence is that the cells reverse direction quasi-periodically. The distribution of reversal times, it's not really peaked. It's sort of more exponential. But every several minutes, every maybe 10 minutes or so, they go the other way. Okay, and if you're interested in sort of synthetic biology kind of clock kind of things, they have a very nice, interesting sort of a relaxation oscillator that they've developed through evolution in terms of these specific proteins and the methylation state of these specific proteins. And importantly, the cell can alter this frequency. So they're always going backwards and forwards, but the cell can turn it off. They can either always go forward or they can go backwards and forwards more quickly, and that depends on signals that they get in from the outside. So they have some level of control of this. Also importantly, because we know things about bacterial genetics, we can control this frequency too from the outside through a combination of mutations in these proteins and the addition of small molecule drugs. We can turn off the reversals, we can make them faster and tune it somewhat. And so we're going to use this in part of our experiments, that we can modulate, perturb the oscillation frequencies of the cells. Okay, so you might want to know what happens when they collide together. So they're flexible. So when they collide together, not only flexible, but they have an asymmetric friction. So it's easier to drag them this way than this way. And so when one cell runs into another cell, it ends up sort of aligning with that cell because they're flexible. And so you naturally, through this mechanical interaction, get alignment of neighboring cells. If they run into each other, they'll want to align with each other. And so if you have lots of them, you can then get nematic alignment of the population, which is what we're going to see later on in the talk. There's a second kind of interaction, and this is that as they crawl, they actually sort of eat a little bit of the surface. We don't really understand exactly how that works, but you can measure in 3D the surface. And so you can see over here, these green spots are where the surface has gotten lower because the cell has gone through that. So they leave a little trench, and then both those cells and other cells will then follow the trenches. Okay, so they align with each other, but they also have this way of essentially following each other. And both of those kind of physical mechanisms are going to be important for the sort of pattern formation that we're going to see. Okay. So I want to talk about many scales that go from the single cell to these droplets. We're going to start by talking about small groups of cells and how those end up wetting the surface. And then we need to talk about the instability that gives rise to dewetting of the surface into the droplets. And in particular, this is an instability they can control, right? If you're a passive material, whether you phase separate depends on all the external variables, right? With temperature being an external variable. If you're an active material, like you could be a bird flock. The birds have brains. The material, the molecules can change their behavior, right? Which means that the material can change its own behavior, which is something that active and biological materials can do that regular materials can't do. Okay, so you might have learned a little bit about flocking. I'm not going to go into the extensive literature of flocking. The most important sort of aspect of this is people usually think of these as basically spin systems. So you have alignment of some subset of your neighbors within the population. So it acts something like a ferromagnet, except that here you move along the spin direction, right? Like if that's your velocity vector as a bird, or your velocity vector is one of these gliding cells. And so this allows you to break symmetries that you're not allowed to break if you're just an actual ferromagnet, for example. And people have done work both on sort of agent-based systems and continuous systems. There's a very nice literature on this. And you can do the same thing with Myxococcus experimentally. So we can break that reversal clock. And so now all the cells only go one way, and they have this nematic alignment, because if they run into each other, they align with each other. They also have this trail following thing. And this is what you see in the end. What you get are flocks, small groups of cells that move coherently together. You'll see that there's really a lot of really fascinating things in this movie. The cells will all move together. You can get circulation flows on the outsides. There's definitely shearing can happen within these. There's some sort of surface tension. You can get groups to merge and leave each other, and all sorts of interesting stuff can happen in these things. So these are flocks, by the way they look. You can also calculate various statistical properties of these things. One of the more interesting ones is that in a distribution of flocking molecules or organisms, if you look at the scaling of the standard deviation of the group size, you find that it goes with a power that's larger than the square root of n. So there's an anomalous scaling that basically tells you that the large groups are more likely to break up than you would have from a thermal system. And this is something called giant number fluctuations. And indeed, if you look in our theories here, so we've scaled out already, we divided by the root n dependence. So the passive response would be a flat line here, and you can see that we have an anomalous scaling, which more or less matches the dimensionality of the system. And so you can predict. And so basically every kind of flocking parameter that you might want to measure that people have discussed, these Myxococcus flocks have the same thing. So this happens when we turned the reversals off. Now, what happened if we turn the reversals on? So here is going to be flocking things with no reversals. And then here I have these wild type cells. I've colored them all in different colors just so it's easier to see. And you can see that there's very, very different phenomenon here. So here you have flocks. They move all around. The group sizes are a little larger in this case, but they break up and they move around, and that's fine. If I run the movie for the reversing cells, what you see is something very different. They basically form static structures that stay there in space. And then there's other regions of space, like over here, where there's never any cells. Okay, so by turning on reversals, we've made it so that you have qualitatively a different phase here. And in fact, if you mix these, they phase separate, right? Because the flocks like crawl away and the reversing cells don't really go anywhere. You can take a histogram, for example, of where all the cells were in space. The flocking cells basically go everywhere. They don't really prefer to be anywhere. Whereas these reversing cells form these things that I like to call streams. Different people this has come up in a number of different literatures, have different sort of language for this. I call them streams. But basically, there's always cells within this stream here, and there's never cells over there. You can look at the fluctuation of the size of these streams over time. So they grow essentially viscously, as if they're wetting the surface as more material comes in. And when you look at those fluctuations as a function of the group size, and you divide by this factor of root n, you can see that there's no anomalous dependence. So these groups don't have these giant number fluctuations. And if you go back to the literature, you can say, well, why were the giant number fluctuations there? Why are they predicted to be there? In fact, they always have to be there in dimension 2 and higher. And so what the Myxococcus have figured out how to do is to make a dimension 1 object. It's a line. And that object doesn't have to suffer from these fluctuations, which means that they can make a population on the surface that doesn't move and break up, that's stable. So this is a way of seeding a location in space that's going to be stable and immune to these giant number fluctuations, even though it's being driven all by these active forces, et cetera, because it's in one dimension. So we spent a little bit of time thinking about where these why this state might happen. And one thing that you really need if you look at simulations of various kinds is you can say, well, what happens if the cells stick to each other? And that doesn't do much except make them sad. But if you make them follow each other through these ditches, which I showed you are there, then you can get these streams, these long-lived streams. Okay, and you can do the same tricks where you look at the histograms, et cetera, to see what happens. So the streaming is important. It's important because it's one-dimensional and allows you to have a long-lived state. And that streaming requires you to have this sort of ditch following, or at least neighbor following. I don't think it has to be ditches. I think you have another way of following your neighbor, but you have to have some way of following the neighbors in front of you. And then you can make this kind of a thing happen. If you look at the motion of a single cell, if it's in a flock, it's basically diffusive. But when it's in a stream, it's more or less ballistic before it reverses. It's like stuck in a line and it's going straight. So that looks ballistic. But as it starts oscillating back and forth, it doesn't diffuse in space. It's basically caged within the stream. And because it's caged within the stream, you get a subdiffusive regime, which tells you that not only is the global state long-lived, but the cells themselves are actually residing much longer in any particular point in space than they would if they were allowed to crawl around either by themselves or with a flock, for example. Now, what I told you about now were cells that don't reverse ever because I mutated the clock that makes them reverse, or cells that always reverse with a very clock-like mechanism because I made a different mutation in the clock. Now, what I want to show you is what cells do when they starve. So what happens in the process of starvation? Well, I'm running around, I'm trying to find food, everything's great. And then I realize I'm starving. But now what I have to do is get together with all my friends so I can start this whole process of wetting and dewetting and all this stuff. So if you look before starvation, did the movies not play? Okay. If you look before starvation, right, what you see is these cells look more flock-like. There's little groups that you can see moving around in different places. And 3 hours after starvation, why is that? There we go. 3 hours after starvation, I basically formed these static streams. So the cells go from a non-reversing state when they're happy into a reversing state that allow them to start wetting the surface. Okay, so here's that 3 hour sort of time period sped up a lot. You can see they're kind of everywhere. And then eventually they form this static structure, and that static structure just spreads. Spreads and spreads and spreads slowly over time. Okay, and you see sort of viscous fingering instabilities. You see lots of things that really look like a fluid that's sort of wetting and fingering out onto a solid substrate. Okay, so we can go from this state, and now a few hours later, we've wetted the whole surface, basically. Okay, so this is a little more zoomed out. You can maybe see some of the little outlines of the cells. I'm not sure how good the video is. You can definitely see kind of the directions. These are holes in the layer. So now rather than just a few cells, we really have a wetted single layer. We have holes in the underlying substrate, a nice monolayer of cells, and we'll play the movie to see what happens now. So you can see there's basically motion everywhere. Everywhere in the bulk, there's motion. The holes open up and close, and what I get are the formation of second layers on top of this first layer. And I don't know if it's in this movie, but you'll get third layers and fourth layers, etc. So once I've wetted onto the surface, now I'm starting this process of dewetting, and the way it happens is via a process of layering. So first I form essentially a monolayer, then that monolayer develops regions of holes where there's no cells, and second layers, and that's how I'm going to build up this eventual fruiting body. Here I've slowed it down and zoomed in a little bit more. Can you guys see the cell outlines? I don't know exactly what you guys can see. Okay, and so there's a couple of things you can take home from this. One is there's basically no jamming. Everywhere you look, the cells are moving, which is pretty fantastic if you ask me. I think a lot of people would thought, and in other systems you see, when you put motile cells at these very high densities, they just kind of jam up. And you can get interesting things to happen because of this. But this really makes a fluid. There's cells shearing all over the place relative to each other and moving all around. You see the nematic alignment is very strong, right? The density here is very high. They're basically close packed, and neighboring cells are aligned with each other. So you have a nematic liquid crystal here that's active. And you also hopefully can see that there are places where the nematic alignment is screwed up. Locations in space that we're going to call topological defects that you don't have perfect alignment of the cells. Okay. So if you've learned anything about liquid crystals, you know that, or if you've just played with like a bunch of matchsticks or something, if you try to stick them all together, you can get regions in the bulk that are nicely aligned. And there's only a finite number of ways. There's an infinite but countable number of ways at which at a point you can screw up that alignment and still have kind of close packing. Okay, and there's going to be different sort of species of these, and we're going to quantify them by calculating the winding number of the director field as we go around these places in space. And because it's nematic, they're not polar. This and this are the same for the cells. So if I calculate the winding number of the director field as I go around the loop, I can either have a negative or positive charge. And that winding number is quantized by half integers, because I can go around once and only turn half a turn. And that would be a half integer, or I could turn a whole turn. That would be a whole integer. You can have 1.5, 2, 2.5, etc. And so in practice, we're only going to see the half, the -1/2 and the +1/2 topological charge defects. If you've studied conventional materials, the energy cost of making a defect goes like the charge to a high power, usually the fourth power, depends somewhat on the details. The same is basically true of active dynamics, and we basically never see point defects that are integer charge or higher. We only see the half integer charge ones. And so I'm going to draw blue, I don't know, triangular threefold symmetric shape here to represent the -1/2 defects. That's this shape here. They have 3 axes of symmetry, and I'll use this sort of comet, this red dot and a red line, to draw the +1/2 defects, which have 1 axis of symmetry, cells that are oriented orthogonal to that axis in front of the comet, you might say, and then you go out along the comet tail, which is the axis of symmetry. Okay, so now we can take our images. So here we have a nice movie of the cells moving around, and we can color code it by the direction of the local director field. Okay, so here red is horizontal, blue is vertical, green is up and to the left, and purple is up and to the right, as described by this sort of color wheel. It's only a half circle because it's nematic and not polar alignment. And then you can play that movie. And what you see is in the bulk, you mostly have gentle gradations or variations of the color scale, meaning that, you know, as you move in the bulk, you can change the director field a little bit. But then you also have special places in space, points, where you basically have all the colors meeting, and those are the single point defects. Okay. You can see that they're kind of connected to each other. There's lots of interesting phenomenon. Holes are a boundary, they can absorb charge. So the holes can have multiples of half integer charge to them. So if they absorb 2 -1/2, they'll have a -1. So as you go around the whole boundary, you'll see a net change of 1 hole winding, for example, things like that. So you can calculate and measure where the topological defects are by looking at the alignment field. Can you see the equation on the bottom? Yes or no? Well, there's an equation on the bottom. Anyway, you look at the local alignment field and you compare it to the average in a small box, which we have, it's like a disk of a micron, I think. And when you do that, you see that the alignment is basically uniform everywhere, except at very specific points, which is the location of the topological defects. So then you can play a movie, you can see the creation of pairs of these. So much like regular charge, you can only create or annihilate topological defects in pairs, except at boundaries. So in the bulk, you can separate a pair and they can annihilate, except. There's a question here. At what point, if you vary size and or shape parameters, do you expect cell jamming? So under these conditions where the cells are able to move very strongly, we have never seen jamming. We can't put them in a closed boundary, or at least I haven't tried to put them in something where it would really increase like the pressure a lot. There's always an open boundary, so I think that maybe prevents things. Where we do see jamming is we can change the surface underneath and where they don't move as well. And if you basically make it so they can't generate very much force, then you can get them to jam. And there's interesting things that can happen there. We think this is a little bit more like what they would see in the wild. We think they move very well, but you can get them to jam if you decrease how well they're able to generate forces on the surface that they can. Okay.
Professor.
Yeah.
We have 2 questions from YouTube.
Go
can
ahead.
I read them?
Mm-hmm.
The first is from Nathan Berkowitz. Can a cell reproduce to create friends? In this case, is it a multi-cell organism?
So the cells do divide, but only when they're eating and happy. So the process we're talking about here is what happens during starvation. So in almost all these movies, if you watch carefully, you won't see cells dividing. because they don't have resources to do so. Whether you want to call this a multi-celled organism is an interesting question. I think I and most of the community would say no. It's a population that does population level things, but I don't think that you would consider, this group to be one organism. I think it's many organisms inside of a population, but that's clearly some sort of semantic debate that I'm sure, involves definitions of things, and one could argue about the definitions if you wanted.
And the other question is, what is the material on the surface where they are digging the trenches in?
So what's the question again?
What's the material that's on the surface where the cells are digging the trenches?
So it's an agarose gel, which is basically a gel made of sugars, sort of. And so we don't actually know, like, if they're eating it or just displacing it. We have not quite figured that out. But they're definitely somehow changing the shape of it. We have some projects working on that right now. Okay, so indeed, if you look at the locations of these points where the alignment field is decreased, you will find topological defects. So this now is an image not of a thumbprint, but of the actual mixo field. We've color-coded the alignment. So green is horizontal and red is vertical. And here you see a -1/2, so the threefold symmetry point defect, and a +1/2, a comet tail defect right there. And so, we can use the alignment field measurement to find the location. We can integrate around and calculate the winding number to calculate the charge. And so now in a movie, we can automatically track the location and charge of all of these defects. So here, what we have is a measurement of what the cells look like. Here we have actually also the height. So I didn't talk about how we make these measurements, but it's a pretty sophisticated microscope that not only tells you essentially the reflectivity of everywhere on the surface, but also what the actual height is with, nanometer-ish, tens of nanometer scale precision. Then we can analyze the height field, and we can define the layers. So here's the 0th layer, the substrate, the first layer, the second layer is going to be in a darker color. And we can look at the location of the topological defects, which I've plotted here in red and blue. So you can see, you can watch defects pairs being born, being annihilated. You can watch defects being absorbed to the boundary of a hole, for example, things like this. Okay, and what we found, which you can see if you stare enough at this movie, here, I'll make it bigger. Oh, it did not advance. All right, is that playing? Good. Okay. So you can see annihilation, you can see pair creation, but what you also might be able to see is often layers are associated with the +1/2 defects, the little red comets, and often holes are associated with the blue -1/2 defects, these threefold symmetry things. And indeed, when holes open up or close, they basically look like triangles, representing the threefold symmetry of the -1/2 defect. People seem to like this movie. I'm going to let it play because you guys look excited. I like movies. So you can look at the motion of these defects. You can calculate something like a potential energy between them by looking at the distribution of distances. And as you might expect, like charges repel each other. So the red and the blue have a barrier between them getting very close. If you sort of think about how you might make two comet tails want to be close to each other in terms of the alignment field, they really don't like to do it. You have to introduce too much splay to the liquid crystal, but opposite charges attract each other and then eventually annihilate each other, just like in electrodynamics. So here we have electrons and positrons or whatever that can propel themselves around and do things. So there's a lot of interesting sort of active fun physics you can do here. I don't think the Mixococcus care too much, but there's some interesting things you can do in terms of active matter phenomenon, thinking about these defect pair interactions. What the Mixococcus do care about, though, is making layers and holes, because their goal presumably, is to make these big droplets. And the way they're going to extrude from the surface is through the generation of layers. And of course, if you make layers, you have to remove the material from somewhere. So that's the generation of holes. And so if you look at, for example, this is going to be a layer formation process. We have the location of this +1/2 defect. It's going to move along and generate a new layer, which you can see here also in the color map. Here we have a -1/2 defect. It's going to slide along to the lower left and open up into a triangular hole, which then kind of gets a little bit rounded over time. You can look at the distance for layers that are formed. Say, how close is a new layer to the closest +1/2 defect compared to the closest -1/2 defect? And you can see that they're always very, very close to a +1/2 defect, thousands of times more likely to be close to a +1/2 than a -1/2 when you form a new layer. If you form a hole, the situation is reversed. You're essentially always at the location of a -1/2 defect and almost never at the location of a +1/2 defect. So it really seems like these defects are driving the layer and hole formation process. You can model this, of course. We aren't the first to have done this. In an active pneumatic liquid crystal, what you basically have, you have conventional force balance. You have applied forces. They're balanced by viscous friction. Our friction is going to be anisotropic so that it's easier to slide along the length of the cell compared to against the length of the cell. So you can relate the friction coefficient, then has to be related to the director field, which is the cell alignment field. Okay. And then what you add is that the stresses and the forces that are generated depend on the direction of the alignment field. Right. So I have this alignment field, it's generating stresses. Those stresses are balanced by these anisotropic frictional forces, and you can let it go and you can solve for the configurations. So for example, you can look at the flow field that you would predict around a +1/2 and a -1/2 defect. For a +1/2 defect, you predict that you have a flow inwards towards the core of the defect. For a -1/2 defect, you predict that you have flow outwards for the -1/2 defect. And if you measure the flow in experiment, you can see we have very good agreement, certainly qualitatively, but even quantitatively, between the patterns of flow that we see in the experiment and the patterns that we predict from this pretty simple model of basically an active pneumatic that has anisotropic friction. You can even quantitatively match the data, which is shown in gray and black here, with the model pretty convincingly. And many of these parameters in the model you can measure independently. So for example, what is the average amount of alignment you have in the bulk far away from the defects? That you can just measure from the experiments, for example. Okay, so it seems like really to get the best flow field matching, we really need this anisotropic friction. We originally didn't put it in the model, but for a number of reasons, it really seemed like you had to have it. So we went out to go measure it. And I'll tell you a little bit about that. This is work in progress. So there's something called traction force microscopy. You might have heard of it. The idea is that you have something that's moving on a deformable substrate. As it moves, it deforms the substrate. If you can measure that deformation and you know the material properties of the substrate, you can back out what forces that moving thing was applying on the substrate. Right? I mean, that's the only way you ever measure forces, right? Is you have a known spring and you measure how much it displaces. It can be a very fancy spring, but all it is a spring, right? Anyone who tells you otherwise is lying. It can be made of light. It can be made of a nanometer scale thing. It doesn't matter. It's a spring. In fact, biology does this too. The way biology measures forces is by making unfolding protein springs and measuring whether they're unfolded or not and how often they're unfolded. So same exact strategy. So we can do this with cells. There's a lot of trickiness. If you're interested, you can look up this paper. Because the cells are small and we want to do it on a small scale, it's hard, but we can do that. And so here, there we go. So here we have our single layer of cells. You can see a -1/2 defect here. We have a hole here. This is the image of the tracer particles underneath this on the surface. And on the right, I'm going to be plotting the traction stress as well as the direction. So the stress is in color. The direction is a little arrows. You can see at the location of the defect, you have a bunch of stresses, things like this. So we take many measurements like this. And what's the first thing we can do? Well, we can measure the distribution of stresses. And then locally, we can tell you how well those stresses are aligned with the pneumatic director. So 0 degrees of alignment would mean that the forces are basically pointing along the alignment direction of the director, and 90 degrees is orthogonal, and you can see we have a strong bias towards orthogonal. So if I have a cell that's pushing this, this creates much more stress on the surface because the friction is higher. If it pushes this way, the friction is lower. Okay, so this is direct evidence for this anisotropic friction. You can then look under the topological defects themselves. So here, the signals are pretty small and hard to measure. So we measure 10,000, tens of thousands of defects, both positive and negative defects. You get a stress field, and then you average them all together for the plus and minus 1/2 defects. And so this is the stress that you get. It's in front of the defect core. Why is that? Because if this is the comet tail, these ones come and run into these guys. And this is where the strong friction is, because this is the forces are pointing orthogonal to the direction of the alignment. And so you get the strong stresses in front of the comet tail, okay, from that anisotropic friction. And that's what causes you to want to form a second layer, because you're running into these cells and they don't want to move. And so there's this process via which you pop up there. And in fact, you can predict these too. These are sort of early calculations, because there's some things in the theory about the forces that we still want to measure to try to figure out better. But you do predict that with the anisotropic friction in front of the plus 1/2 defects, you get a stronger stress, and you also get stresses orthogonal to the arms of this blue threefold symmetric thing, which is what we measure in experiments also. Okay. So the last part of this then, we've said that we go from a single layer. We've talked about 2 layers. Now, if we watch how it goes from there to building the whole dewetted droplet. So here we're starting at the point where we have, this is the underneath substrate. This is the first layer. Here's a second layer. You can see we've started to dewet even more, and you still see layers here on the edge. And as you let this movie play, you can see that the instability really is layers growing upon layers, growing upon layers for long times. And in fact, if you let it go longer, this is at lower resolution now, you still see layers in the basic substrate, even as things grow much taller, and you get many, many layers that end up forming things that look really like round droplets. Right, that are really quite round and smooth and have a contact angle, except for the very first couple layers on the edges. Okay. So how do the cells make it so that they prefer this state to this state? We experimentally can tune the cells so that they basically are very happy in a single layer, and they make holes and they make new layers, but about the same probability. And the thing just kind of sits there making holes in layers until it runs, just sort of runs out of fuel. And then the experiment kind of dies. But the cells have to somehow favor this dewetted state. They have to favor the layer formation over the hole formation process when they're starving, so they can end up making this mound that they sporulate in. Okay, and so we can think of this as some classical dewetting phase diagram, where your stable states are essentially wetted layers, and dewetted droplets. There's some sort of phase boundary at the binodal. You can have coexistence inside this phase down boundary, both droplets and layers at the inflection point in what would be the free energy diagram. In a passive system, you'll have a spinodal point, etc. Now, some of you may be saying, but why are you showing me this thing? What is, this is a bunch of self-propelled moving bacteria. Why should you expect that you can build a phase diagram that looks like this? And the answer is you should not. There is no reason a priori why this would have to be true. But what I will show you is that it is true. Okay, so not only does this active material look like a fluid just when you watch it, it actually seems to behave very much like a fluid, even though it has weird properties and it's self-propelled, much more so than you might have imagined. So here we're going to take Myxococcus and put them down in a very thin layer. It's a very low density. And I'm going to play a movie, and you probably can't even tell that it's playing. Maybe you can. Maybe you see the gray kind of doing a little bit of something. If you stare hard enough, you might see a little dark gray thing kind of moving around. And that's basically a flock. It's really small, hard to see. So this is staying in a wetted phase. Okay, you're not seeing a dewetted phase. Now I'm going to take those same cells prepared the same way, but now there's more of them. So the layer is thicker. And I'm going to start the movie. And what you're going to see is that little black droplets are going to form at random places in random times throughout the field. Okay, and you'll see even more interesting things if you're pretty eagle-eyed. Some of them dissolve, so they form a droplet and they disappear. And in fact, the little ones seem to dissolve, and the larger it is, the less likely it is to dissolve. And the litter ones, if you look really close, which you can't see in this movie, you can see a flux between the little ones and the big ones. And it really looks like conventional Oswald ripening with a regular phase separation process that you would have predicted for a passive material. Okay. Now you make it even thicker. And I'm going to play the movie. And I want to tell you what's going to happen first because it happens really fast. Rather than getting nucleation of droplets that happen at random places at random times throughout the field, everything is going to phase separate at once. First on small scales, and then it's going to coarsen. So there it goes. Right? So qualitatively different phenomenon. You still get droplets in the end. But this looks much more like spinodal decomposition, where small fluctuations everywhere in the field are instantly amplified. And then whatever the resultant pattern is has to coarsen over time. You can study this coarsening. So for example, you can calculate the structure factor by taking basically Fourier transforms of these images in the movies over time. You can look at the structure factor. You can see that we can collapse the structure factor from different times based on a single number, the position of the peak. You can see the shape of the structure factor is self-similar, as you would expect for spinodal decomposition, and that the size scale undergoes a power law as the length scale grows and coarsens over time. And so, in fact, you can predict these scaling exponents pretty well. They match simulation theory very, very well. There's actually an interesting discrepancy with some analytical theories that I'm not sure has been worked out yet. But this really is supposed to be more like a quarter or 0.3 and not 1/3, as you might have seen in some classes if you went through these sorts of things. But it really looks like a conventional dewetting material, even though it's being driven by these thermal motions. So now, clearly, temperature is not the important parameter here when thinking about whether you're going to favor dewetting or wetting, moving around on this phase diagram. So the thickness of the film is going to be one axis of the phase diagram, but temperature is not going to be the other axis. What you want is something that quantifies essentially the fluctuations in the system. And so for many active matter systems where you have self-propelled objects, the thing that quantifies the fluctuations is called the rotational Peclet number. And I'm using the inverse rotational Peclet number because that has the same sign as temperature and it will make the phase diagram look normal to you. But it's just the ratio in a conventional system, right? If I have a river flowing down and I put some dye in it and I ask how does diffusion compare to transport, that's what the Peclet number tells you. And so it's a ratio of the advective stuff to the diffusive stuff. And in terms of the parameters for the Myxococcus xanthus, there's this, which has to do with how straight cells normally move, if they're just by themselves. And that we can measure, right? If they're trying to go completely straight, there's a little bit of wiggliness to their paths. And that's, that's some, stochastic thing, and we can measure that. Then there's the reversal frequency. And if you change the reversal frequency, you change effectively how diffusive the motion is, right? So you can make it look much more diffusive by keeping this constant and having it go back and forth more. And then there's the speed of the cells, which is basically like the advective transport. Importantly, the cells can change this and can change this based, on their whim. They can measure how much food is around their experience, and they can change these numbers. Now, equally as importantly, I can also change those numbers because I have mutants and drugs that allow me to change the reversal frequency and the speed. So I'm going to use different combinations of mutants and drugs to play with the reversal frequency, to play with the speed, to put populations of Myxococcus xanthus at different Peclet numbers. And when I do that, I see that at different densities or different thicknesses on the surface, there are Peclet numbers in which I will not phase separate. I'll remain wetted on the surface. And then there's Peclet numbers in which I will. So I don't phase separate, or I do phase separate, I don't phase separate. So I can go into the coexistence regime either by changing the density on the surface or by changing this Peclet number, by either making them go faster or by making them reverse less. Okay, and I can do it both ways in the experimental system. Now, what does Myxococcus do when you starve it? So now this is like the wild type, so the regular organism. I take away all its food, and what it does is it goes faster and it lowers its reversal frequency. This causes this inverse Peclet number to go from a value of about 2 down to a value of about 0.2, so about a 10-fold reduction. And it goes from about up here before starvation down, crossing this phase boundary right after starvation. So this is a material where the molecules themselves can change their temperature, right? They're like what would be the kinetic energy, their motion, based on cues from the outside. And if the whole population basically does that, they can affect a phase change of the population. The population can go from wetted to dewetted, all because they decided to do it themselves. And that's what I think is super cool about biology and living systems. So I think with that, it's been exactly an hour or something. So it's timed pretty well. We had a few questions. I'm happy to take more questions. The organism I just told you about forms these things on the forest floor of many, if you go outside in many wet environments, they look like this. Some of what you thought might have been fungi are actually Myxococcus xanthus. All of these other patterns are also formed by relatives of this bacterium that clearly make much more complicated, interesting patterns. We don't have them in the lab and can't do as many things with them, but they make interesting patterns. And so they seem like they might be fun to study. So, okay, I will stop there and take any more questions that may have come up.
Thank you, Professor Shaevitz. It was a great talk, really interesting. So I'll move on. I'll prioritize the questions from YouTube because we're still broadcasting.
Mm-hmm.
So we have just one question at the moment from Nathan Berkowitz. What is the evolutionary reason why the cells are in constant motion? Is it to hunt for food sources, and do they compete with each other for the food?
So I think almost everything they seem to do involves motion until they sporulate. So they hunt by moving. When the they need a population to make high concentrations of antibiotics, but they also need to get themselves closer. So those waves I showed you in the beginning, I think part of their function is to sort of mix the bacterial population so that everybody gets a chance to absorb whatever nutrients from the things that they killed. Then clearly this phase separation, this dewetting transition relies on them moving, right? And then they sporulate and then they stop moving. Whether single cells like this compete for food, I mean, yes, they're always competing, but they're also in the same population. So this gets into more of these semantic things about how much they're actually competing with each other versus not competing with each other, and what the definition of kin is and species is for bacteria, because they're sort of strange this way. But I hope that answered the question somewhat.
Another question is, if you take the food concentration as the order parameter, what kind of phase transition is it? First order, second order, etc.
I don't know. It's a little tricky because you have to integrate the history, right? So if you just give it a bunch of food, it will eat it all and then undergo the dewetting transition, right? Because it runs out of food. So then you could imagine, okay, well now I'm going to titrate different amounts of the food, and then I have to understand this integral about how much food is left to figure out when it happens. And I think that seems a little hard to me, because even so, like you can change how long it takes them to de-wet depending on how strict you are about starving them. So if you work really, really hard to get rid of all the food as quickly as possible, which is not very quick, unfortunately, just experimentally, basically this process kind of happens right away. If you're more sloppy about it, like if you put them on a substrate that just has a little bit of residual sugar, it takes much longer for them to de-wet because they have a little bit of food, so they eat it, they're kind of happy for a while, and then they decide to undergo the whole thing. So I think this is a hard question to answer. That was a long-winded answer, but
Another question, during starvation, do some of them die? And if so, are they eating each other?
yes, and yes. So during starvation, many of them die. Only a fraction of them end up being spores if you go all the way through the process, and that fraction depends on different things, but you know, as many as 70% might be able to do it. Many of them die. It is definitely true that Myxococcus can eat other Myxococcus. In fact, in one of the moves I showed early on, I didn't point it out, but there's sort of a dead cell there. It looks like a blob, and the other cells just come and sort of rub against it, and the blob slowly disappears. So they will happily eat their friends. How much of that happens in the fruiting body, though, is a different question, because at that point the cells, like once they've committed to sporulate, I don't think they're doing anything else besides trying to build this spore coat and stuff like that. So I'm not sure how much of that is purposeful, but it might be that along the way some of those nutrients are absorbed, and then you know the other cells get to use those.
Okay, so just let me check if I have any more questions there. And okay, I think we can end the broadcast now and move on to the discussion here at Zoom.